my name is Nabihah Tayob. I am an assistant professor in the Department of Data Sciences here at Dana-Farber. One of the key areas of my research interest is biomarkers. And so today I'm happy to be leading this training session on biomarkers in cancer research. Just to kind of a heads up, we are in a meeting format for this the Zoom call. So you can unmute yourself at will. I would request that you just be careful and don't unmute yourself when you don't intend to. But feel free to stop me if at any point anything is unclear, if you have any questions. We can have any kind of, I would say, like, if anything's unclear as that is relevant to the content as we're discussing, then just stop me and ask your questions. Unmute yourself and ask your questions. I would say more kind of like discussion philosophical things. Maybe we keep those to the end. I'm going to also do a break because it's 2 hours. I think that's a long time to be sitting in one place. So we're going to do a break at about the hour mark. So if you have any of your questions, you can ask them during that break time. What other heads up? Yeah, so just feel free to unmute yourself, open your video, and ask your questions. And we're going to go ahead and get started. Okay, what is a biomarker? We have an NCI definition of a biomarker that is a biological molecule that is found in blood, other body fluids or tissues that is a sign of a normal or an abnormal process or of a condition or of a disease. A biomarker may be used to see how well the body responds to treatment. Sorry, I'm gonna please move this so I can see my screen. Or disease, for a disease or condition, also called a molecular marker or signature molecule. So this is the official NCI definition. When you go onto their website, this is what you see. I would say that, to me, I kind of think of a biomarker a little bit more generally as basically anything that will allow the classification, anything you can measure in a patient that will allow the classification of the patients. And in the context of cancer, we can think of biomarkers for risk stratification, early detection, diagnosis, prediction, prognosis, treatment response. I would also argue that we don't just necessarily say something that's measured in blood or fluids. I would also say you could have biomarkers from imaging studies. So for example, there's an entire field of radiomics whereby you can quantify what you're seeing in an image in various ways. And so once you've done that quantification, I would argue that those are biomarkers too, that you can use for these kind of contexts and analyze. So I take a very general approach for what is a biomarker. A biomarker is something that we measure in a patient and we use to make decisions about the patient, whatever the context of their in the spectrum of cancer. So across the cancer continuum, we have biomarkers at various points. Before the cancer onset, we have risk stratification biomarkers. So who's a high risk patient, who's a low risk patient. Once the cancer is onset, but before we diagnose it, we have early detection biomarkers, and that's a particular interest of mine. So are there other things we can measure in a patient to detect the cancer earlier? Once we can also have biomarkers for diagnosis. So can we think of things in the patient that we can use to diagnose the cancer? So maybe you want to avoid having to do biopsies, and are there biomarkers that can avoid biopsies in some patients? Once you have a cancer diagnosis, then we have biomarkers for prognosis. How well is the patient going to do? Maybe if they have a particular marker, maybe that tells you something about how their disease will progress. Predictive biomarkers are biomarkers that tell you about their potential response to treatment. That's the context we're using it here. Monitoring, maybe once you've completed your your initial treatment for your cancer, you might be monitoring the patient for a cancer recurrence. So so you could have biomarkers for that. We also have, maybe once you have a cancer recurrence, then you have a prognostic biomarker. Maybe it's the same marker, different biomarkers. So throughout the cancer continuum, there's biomarkers that we can use at various time points The key is to understand the context in which you're using the biomarkers, and that helps us evaluate the biomarkers for their clinical utility. So I understand that we actually have a pretty general audience today. So that's great because I think it's very useful for everybody to have this information. I did kind of tailor this talk towards statisticians. So some of this part is that, but I would just actually say that for even anybody who is a clinician, basic science researcher, it's actually really important to understand the statistical perspective, because when you work with us for designing the studies and analyzing your data, it's important to kind of think of what are we thinking about. So from a statistician's perspective, what do you need to consider before you receive any data on this biomarker? Oh, sorry, there was a question in the chat. What's the difference between a prognostic biomarker and a predictive biomarker? Essentially, a prognostic biomarker, and we'll get into this some more when we go into clinical trial design, but prognostic biomarkers do not allow you to make any treatment decisions. They are about how the patients will, they progress. So let's say they have a particular marker that gives them high risk of developing worsened disease than others. Without the biomarker, that's a prognostic biomarker. But a predictive biomarker specifically helps you to guide your treatment decisions. So it helps you tell you which treatment is potentially preferable if you have this biomarker present or absent. But we'll get into that a little bit more in the second half of the training session. So before you receive any data, I think there's some key things to understand about the biomarker. So you want to know, what is the phase of this biomarker development? Is this a novel biomarker that hasn't been studied before? So this lab has been come up with the potential biomarker that they've developed in-house. It's brand new. They developed the assay for it. They developed the technology. They've got maybe some preclinical data. They've got some preliminary data. That is a very different context from one that maybe is a biomarker that is standardized, FDA approved. It's just being used now in a different context. So you want to understand, like, the biomarker itself. Is it something that's brand new, that there's still like a lot of development, a lot of, maybe you don't understand completely, maybe whether it's specific to a particular cancer, specific to some scenario. You might have some preliminary data that it's interesting, but it's still a very novel biomarker, versus one that maybe it's known, it's a well understood biomarker. Now you're just looking at it in a different context. And maybe it's useful in another context. And so it makes a difference because it helps us understand, like, how rigorous is the assessment of that biomarker, because that is important to understand. The next question is, do we have any preliminary data on the intended use? So has it actually been studied in its intended application? Maybe they had a retrospective study, they went back and did the measurement of the biomarkers. So it is retrospective, but it's actually in the context it wants to be used. So for example, if you have a cohort study and you have a potential early detection biomarker, you've got these specimens, you went and tested it in these specimens, and now, so you have it in the intended use population, your high-risk population, you've measured the biomarker, you understand its performance in those who developed cancer, those who haven't developed cancer. You could also, though, only have in the very discovery, the preliminary data is at the more of the discovery level. So it's not as in quite the intended use population. And so you're maybe this, so the reason we want to know this is because we want to know what's the next study that we need to plan. Do we need to plan a rigorous validation study where the idea is to get information on its clinical utility, and it's in maybe a higher level of rigor? Or if you only have very discovery level data that was maybe not in the right context, then we don't want to spend, we want to design a study that gets us to our target population, but maybe we can't afford to do the kind of the level of rigorous clinical validation that we might need. So I'll get into a little bit more, but the idea is as we're doing these analyses and developing these biomarkers, you want to start off getting a discovery, initial signals of utility, and then slowly increase the rigor, because as you increase the rigor, you increase the expense of doing the study. You increase the number of patients you require to do the study. So you want to a certain extent, there's an argument for building it up like we do in clinical trials, where the phase 1 is smaller studies, phase 2 a little bit bigger, phase 3 you do these large trials. You don't want to immediately go to a large trial when you only have very preliminary data that's not even in your intended use population. So you want to have that kind of consistency and that in that slow development, because then you're more likely to actually move the biomarker forward and also not waste resources. The other question is, how robust is the assay being used to measure the biomarker? I would say before you get to a study that tries to basically do any kind of validation, you need to do, if it's an in-house assay being used to measure this biomarker, you need to do analytic validation of this biomarker. You need to understand its reproducibility, and I'll go into more context. But the biomarker itself should not be as much of a source of variation. If the biomarker itself varies a lot with batch effects, or how you measure how you measure it, like are you using retrospective specimens, are you using prospective specimens, all of those kind of things. If it's going to vary a lot with all of that, then that's going to affect your clinical validation. So you actually need to have, once you get to a study that's large enough where you're actually trying to do clinical validation, your assay and your biomarker itself have to be locked down to a certain extent. You have to have good data on that. And that actually can be very challenging sometimes. These biomarker these companies that develop biomarkers spend a lot of money doing this work. So it's a little bit of a tricky balance. Like, you want to move your biomarker forward, but then you have to keep in mind. And then if for example, let's say it's reasonably robust, but it's not actually like, at the level of like an FDA approved assay, there are ways then that we can think about designing the study to improve the rigor when we actually do the validation. The key though is to think about what is the target population where your biomarker be used, and what is current clinical practice. So as a statistician, for us to be able to design the study, we need to know, where do you want to use this biomarker, and how do you want to use it? What is the current clinical practice in this target population? So for example, let's say you have prostate cancer. Let's think about prostate cancer. So you have an early detection biomarker, PSA, and then there's other biomarkers as well, but we won't go to it. Let's say you have a positive screen in this patient. The current practice is that everybody goes for a biopsy. Maybe you have this new biomarker where you know that you're sending people who don't have high-grade prostate cancer for biopsies, and you want to avoid that. So you need to understand the current clinical practice, and then you want to say, how will this biomarker be used to change the current clinical practice? Because then that's how we need to study the biomarker. We need to see what is the intended use, and how is it going to change the current clinical practice? Because that'll help us understand, how do we measure its effect, and how do we come up with thresholds for, yes, this is a useful biomarker or not. So when we're initial discussions, we want to know, what is the target population? Because that's where you actually need to study the biomarker. The biomarker must be studied in the population you want to use it in. What is the current clinical practice in that population? And what is the intended use of the biomarker? How will it change that current clinical practice? So those are really key questions when you're designing and thinking or analyzing any biomarker data. Okay. Just to go into a little bit about the analytical performance of the biomarker assays. So some key things that you want in your biomarker assay once you're getting to the point of validation studies. Reproducibility. does the assay give the same result each time it's run on like the same specimen? So for example, like when you run it multiple times, are you getting the same result? Robustness. Does the assay give the same results when it's performed in 2 different laboratories independently? So if you perform it at a lab in, let's say, Dana-Farber, you perform it in a lab at the Brigham, are you going to get the same results? If you're going to perform in a lab in California, are you going to get the same result? Limits of detection. This is actually this is really important once we get to the evaluation. What are the upper limits and lower limits of detection for this assay? Because sometimes it actually can make a difference on how we evaluate the biomarkers, because if, for example, you have your lower limit of detection is too high, so you can't actually detect these very low levels, and that's actually the threshold where you need to be, like, that's where the biomarker is important, and that can actually be an issue. And so understanding the limits of detection, but also then in its context that you want to use it, how those limits of detection affect the performance of your biomarker. So I kind of think of the different like levels of analytical performance of, so you could have research assays. So these are developed in-house. They can't be used to report results to patients, and they can be varying levels of reproducibility, robustness, and things like that. So these assays, though, then are ones where we need to be a lot more careful about how we do the evaluation of these biomarkers, and kind of if it hasn't been developed to the level of rigor, then how can we introduce things like blinding, randomization to introduce more rigor as we're doing the evaluation, so that we have more confidence when we see the results. You could also have an FDA approved assay. So let's say you have a biomarker that's been previously developed for another context. Now you're using it in a different context. The biomarker assay itself is FDA approved maybe. So it's actually gone through all this level of rigor. You're now just using it in a different context. You probably still want to think about, like, in your context, do these criteria still apply? But you don't have the same, I would say, level of concern that you would have if it's a completely in-house developed assay that hasn't gone through this level of screening. And then you also have clear approved laboratories where I kind of think of these as almost like in between. They have had some level of rigor and assessments, but it's not obviously the level of an FDA approved biomarker. So when we say, I keep going back to this, but the evaluation of the biomarkers needs to be for its intended clinical use. So it needs to be evaluated in the target population with the intended clinical use. Another thing to be kind of thinking about is, so for example, sometimes we can do the initial discovery in a different source. So for example, you could have tissue biomarkers to identify certain targets that you're interested in, but your intended clinical use is to have the biomarkers be measured in the blood. So you want a non-invasive test to be able to do the biomarker evaluation. Then once you get to the validation stage, it does actually you need to transition to the blood biomarker. You can't keep doing the tissue biomarker. You actually need to transition to the intended use of the biomarker. Once again, target population, who are we going to use this biomarker on? So just a couple examples to ground this. So for example, I do research in early detection of HCC. Our target population are patients with cirrhosis. Those are patients at high risk of HCC. They are recommended to undergo surveillance. So we want to do early detection in that population. Another example is diagnosis of minimal residual disease in those who have completed their neoadjuvant chemotherapy. So the target population is patients who have completed their neoadjuvant chemotherapy, and we want to be able to use, look for minimal residual disease in that population. So do we have a biomarker for minimal residual disease? I also think kind of to go back to the idea of like, how are we going to use this biomarker? I think there's like kind of 2 broad categories, a rule-in test and a rule-out test. So a rule-in test and this relates to what's current clinical practice. So a rule-in test is when the current clinical practice is no one gets the intervention. So we then want to use the biomarker to decide who should be getting some kind of a clinical intervention. So currently no one gets it. Now we want to decide, okay, who needs to get this clinical intervention? And in that context, we want to control how often we have clinical interventions in those people who don't need it. So false positives. We use the biomarker and we say, you need a clinical intervention, but actually they didn't need it. So when it's in the context of no one getting it, we want to say, how many people do we falsely give a clinical intervention to? That's what we often want to control, a false positive. With a rule out test, that's a context where, let's say, everyone gets the clinical intervention in the current practice. So for example, my prostate cancer example, current practice, let's say, is when your biomarker screen is positive, everyone gets a biopsy. But we want to use this new biomarker to decide some people do not need the biopsy. So we want to control, we want to use the biomarker to decide who doesn't need the biopsy. So in this context, we want to make sure everybody who has cancer and who is high risk, who has high grade cancer, we want to make sure we have high sensitivity. All those patients still gets detected, but we want to control how often we don't do a clinical intervention in those who require it. So how often did we have a false negative? We said, okay, no, you don't need a biopsy, but actually you had high grade cancer and we should have sent you for a biopsy. So that's kind of what I say about what's the current clinical practice and what are we using this biomarker to do. And I think like these rule-in tests and rule-out tests kind of are like nice broad categories that help us think about this. And the decision of what's considered a sufficiently useful biomarker involves trade-offs. So it involves understanding the consequence of a false positive and a false negative result. So for example, with early detection of HCC, you send patients for, when they're undergoing screening, let's say they have a false positive, your biomarker says, okay, this patient potentially has cancer right now. The consequence of that is they go for a CT and MRI. So it's an imaging study, there's stress, there's all kinds of like potential side effects of like, the how long it takes and the additional maybe follow-up tests, but it's not a biopsy. It's a less invasive study. So it's a less invasive follow-up of the false positive. So we can be a little bit more liberal in that. We maybe not going to be as concerned each time. Ovarian cancer, in ovarian cancer screening, when you have a positive screen result, you have surgery. That's a huge consequence. So you need to be really careful about false positives in that context, because a surgery is a major consequence. It's it's the opposite of non-invasive. So in the context, it really depends on the context to understand the trade-offs and what are the consequences of these false positives and false negatives. Okay, so guidelines for biomarker development. There have been a number of, like, in the various contexts, the proposals for how we actually develop these biomarkers. Like I said, I've been involved in early detection. So I'm going to start there with some of the work that's been done to kind of add rigor to this process. I think that although this is specific to early detection, and I'll go through another examples, it's actually the ideas and the concepts apply regardless of where you're applying the biomarker on the cancer continuum. So for example, in the context of early detection, the NCI's Early Detection Research Network came up with the 5 phases of development. So the idea is you increase the level of rigor required to move to the next step. So you start out in your preclinical exploratory phase. So in this context, you're maybe using convenience samples. You're using samples, you're using studies that you had available. You might not have early stage cancers, because that's quite key. You might only have later stage cancers, for example. So These studies are not very rigorous necessarily. It's just a matter of, can we come up with promising directions? And the reason the base is so broad is often we have lots and lots of these studies. However, the next level then starts to move to clinical assays and validations. So you need a clinical assay. So you need an assay that has, that's in its intended use. So let's say if you're going to use a blood test to do the biomarker evaluation, then it needs to be in the blood, and it needs to have like a certain amount of rigor, reproducibility evaluations, things like that. And in the context of early detection, we're saying, okay, we've moved from just potential exploratory to, can it actually distinguish those with the disease from those without disease? Phase 3, then we become a bit more rigorous and more expensive, because now we're going to a cohort study. So we have a retrospective longitudinal study. We enroll patients who are in the target population. We follow them up, and we collect specimens at whatever interval is necessary. And then retrospectively, we go back and evaluate the biomarker and see how early prior to their clinical diagnosis did the biomarker indicate a screen positive. So this is now a phase 2 study, is often like a case control study. So it's a single time point, collect your specimens, do your evaluation. Retrospective now is a cohort study. So you're increasing the number of patients, you're increasing the cost of these studies, but you're also increasing the rigor, because now you're saying not only can this biomarker detect disease, it can detect disease in a preclinical status. So this is getting closer to its intended use. The intended use of an early detection biomarker is to detect preclinical disease. But that's the first level of the intended use. What the key intended use of an early detection biomarker is to detect preclinical disease, intervene at an early stage with a treatment that then improves mortality. So when we get to phase 4 and phase 5, our goal now is to get towards evaluating the biomarker for its goal, which is, can we use early detection biomarkers to improve mortality for that cancer? And so, but prospectively, when we get to phase 4, we're prospectively applying the biomarker and using it to make decisions. And then phase 5, for example, is like a randomized trial. We have patients either screening with the biomarker, without the biomarker, and is there actually an impact on mortality? So those are obviously very expensive studies to do, but that's the key intended use. So you can see how we move from these very, it's in the, as much as close as possible to our, promising direction, lots and lots of studies, moving up to the top of the triangle where we're doing trials, clinical trials to evaluate on the biomarker in its for its intended use, which is, can it impact mortality? So this systematic approach is more likely to increase, it's likely to increase the likelihood, its approach to increase the likelihood of identifying a biomarker that can impact overall survival rates. So by moving through each of these phases, at each point, the goal is to get close to sieve out your potential biomarkers and find ones that are actually going to have an impact on survival rates. You can also think about these studies as phase 1, phase 2, phase 3 are actually retrospective studies. But once we get to phase 4 and phase 5, then we're doing prospective studies. So we want to really understand a biomarker well before we start using it to make interventions. You don't want to do you don't want to move from a preclinical exploratory study to a prospective screening study, because you're going to intervene potentially without fully understanding what you're doing. And 2, these prospective studies are actually, they're expensive, and they have consequences. So you actually want to fully understand what the biomarker is. And so that's why these series of retrospective and before moving to prospective studies have been proposed. So just in terms of these retrospective studies for biomarker evaluation, so the probes design has been proposed for early detection biomarkers, but it actually, I think, like I said, applies in other contexts. So prospective specimen collection, retrospective blinded evaluation. So some of the key principles is that you've collected your specimens in your intended clinical application. So you've collected your specimens in the target population for your biomarker use, and let's say you've collected them actually prior to identifying the disease status. So let's say if you're doing a biomarker for diagnosis, you have your population of patients where you want to use the biomarker to diagnose. Let's say they have, let's say you see something on an imaging study and you want to be able to decide what to do, and you have a biomarker that you want to use to make the diagnosis. It'll be everybody who had an abnormal finding on their imaging study, and then you would then roll them into your study, collect the specimen of interest, and then figure out, do they have cancer or not? So your specimen has been collected when you do not know the true disease status of the patient. Retrospective blinded evaluation, so outcome of interest determined after the specimen is collected. You also have a standard procedure for collecting the specimens, processing the specimen, storage, and retrieval. And this is where understanding your biomarker itself, your assays, is key. You want to know what factors actually affect your assay. Like how does a specimen need to be collected, stored, in order to have that level of rigor and reproducibility. And I have been in studies where, for example, the reagent used in the assay makes a difference. And so you end up running these assays and then you find these like major batch effects. And it's a huge problem. So after you've finished the study, so understanding as much as possible before you start the study is really key. Obviously, we can't control everything in life, and at some point we just move on and we just try it. But as much as is actually feasible should be done. Storage, how old are these samples? Like how long have they been stored? Can sometimes make a big difference in these evaluation of these biomarkers and the assays. So in these probe-compliant designs, the idea is to have a standard procedure. So all the specimens have been processed, stored, retrieved, all in the same way. So you're controlling as much as you can so that when you see the differences in the biomarkers, it's not due to these factors, it then can be attributed to differences in disease status. And that's another reason why you want to do this after the specimens collect. You want to evaluate your outcome of interest after the specimen has been collected. You don't want any biases to creep in where like the cases were treated differently. Patients with cancer were treated differently from patients without cancer, their specimens, because then you've completely confounded any effect of the biomarker if there is any effects. So if you have, for example, I would say even an FDA approved assay actually, because nothing's perfect. Blinding and randomization when you're running your assay and measuring the biomarker is key. It just ensures that, so blinding, by blinding, I mean the person running the assay has no idea what specimens are, the status of the specimens are. So they are completely blinded to the case control status, whether they're from the same patient or not, everything. It's for them, they won't be able to tell any of the specimens apart. Randomization, we often use, like, let's say you run them in batches because they can only run at a certain number of time. Introducing randomization means, again, you don't have, so for example, all the cases measured at the beginning, all the controls measured at the end. We kind of make sure that we do block randomization, for example, to make sure that if there are any effects that are, or time effects, processing effects, that we've actually, there's no bias in case control status and these factors. And so I would argue that for all assays, we should be doing it like this. That is the highest level of rigor. And it involves working with your statistician before you run the assay. And then it's a little bit of work to blind it. But I would say that if you've done this blinding and randomization, then when you go to analyze the data, you have more confidence in attributing the result to the actual intended use, and less concern about biases that could have resulted. And how will the biomarker be used with existing biomarkers and clinical information? So this kind of gets into what I was saying about what's the current use, and how does this biomarker fit in with the current practice. And we'll kind of go into a little bit more about this later. But thinking about this before the study, at the study design stage, is key. So those were kind of some guidelines that I think, although they were developed specifically for early detection biomarkers, I think they actually apply throughout the context for any type of biomarker. Some other guidelines that have been developed for prognostic biomarkers, the REMARK guidelines. So I'm not going to go through, you can see here, there's like 20 different checklists in the checklist of like, these are the things you need to describe. The REMARK guidelines were essentially developed in order to improve the publication of results. So by making sure that people fully report on how they evaluated the biomarkers, the specimen collection, all these factors that we've been talking about, by making sure that it's included in the publication, it allows us to truly evaluate whether the biomarker has the clinical utility it's proposing. And by setting out these kind of guidelines for reporting, I think it also then elucidates important aspects of the biomarker development and analysis that actually should be considered during the design stage. And so I would say that when you go through this, I'm not going to go through in detail because it's everything we've been discussing so far, there are actually common principles for studying biomarkers across these different contexts. So all the things we've been talking about is actually still applicable no matter what context. Now we get into a little bit of statistics and some formulae, but I think it's quite useful for everybody to understand these. I'm assuming no prior knowledge here. So for people who know what an ROC curve is and things, this is probably going to be quite basic, but I think that there are plenty of people who haven't seen these before. And so going into some of the details are important because often we gloss over these. So statistical evaluation of biomarkers. A common tool we use is for understanding classification. And so the text, oh, and sorry, I have references here, and then I'll provide a list of references at the end as well. The book that I learned how all of this to learn this all from is Margaret Pepe's Statistical Evaluation of Medical Tests for Classification and Prediction. I find this a very useful text. So if anybody wants to learn more, I would definitely purchase it. So some notation. We use the letter D to indicate a disease status. So when D is 1, the patient has whatever disease. 0 is disease-free. Disease is used in a very specific, a very general context, whatever, outcome, condition, whatever. So binary outcome, yes, no, do they have the disease or not? Or do they have the condition or not? The Y is denoting a biomarker itself. So another is still sitting in thinking of the simplest case of a dichotomous biomarker with 1 if it's test positive and 0 if it's test negative. So we can have then a 2x2 cross tabulation of how does the disease status and the test results interact. So patients basically can be categorized in this 2x2 table as either true negatives, they don't have the disease and their test is negative. True positive in this bottom corner, they have the disease and their test is positive. And then the mistakes we would make is if, let's say they have the disease, but their test is negative, we call that a false negative. And then if they don't have the disease, but their test is positive, we call that a false positive. So once we're evaluating this biomarker or the test, this is how we can separate patients out into what is their status relative to their true disease status. Because the idea is this test is telling us something about the true disease state. So when we get it right, we get true negatives and true positives. When we get it wrong, it's a false negative and a false positive. Okay. So when we have a binary biomarker, we can think of summary measures of its performance in 2 kind of categories: classification probabilities and predictive values. So classification probabilities are used to measure the accuracy of the biomarker. So to what degree does the test reflect the true disease state? So it's things that you might be familiar with: sensitivity, or also called true positive fraction. So that's the probability that the test is positive given that you have the disease. So the straight line means conditional probabilities. So among those who have the disease, what's the probability that your test is positive? Specificity, true negative fraction, is among those who don't have the disease, what's the probability that your test is negative? So that's your true negative fraction. Your specificity tells you how well can you use, how does the biomarker perform in ruling out the disease in patients. Sensitivity is how well does the biomarker perform ruling in patients, kind of getting into our rule out, rule in that we talked about earlier. And then also we have in specificity and false positive fraction are just one minus each other. So in terms of like when we're evaluating it, sometimes we talk about specificity, sometimes we talk about false positive fraction. They're interchangeable, just one minus the other. On the other hand, we also have measures called predictive values. They are actually used for clinical prediction. So I would think of like classification probabilities is thinking about the biomarker in the context of, okay, I'm studying my biomarker. I want to know how accurate it is. I want to know how it's performing. Predictive values is actually more relevant maybe for a clinical, from someone thinking of it from a clinical perspective. If I do this test on the patient, how likely is disease given the test result is positive or negative? So it's changing your perspective. And we'll go into, it does change, it does affect, it does have some key differences in terms of evaluation that we need to be careful with though. So positive predictive value is, if my test is positive, what's the probability that I have the disease? So I have a positive test, what's the probability that I actually have the disease? Negative predictive value, if my test is negative, what is the probability that I don't have the disease? So depending on the context, we might want to think about the positive predictive value or the negative predictive value. These are all related to each other though. So for the statisticians in the audience, the way you get these relationships is using Bayes' theorem. But essentially, you can see here, so the positive predictive value is actually a function of your true positive fraction, your sensitivity, and your false positive fraction, one minus your specificity, and rho. Rho is the population prevalence of the disease. So the reason why it's important to understand this is because the population prevalence of the disease is needs to be reflective of the what's the actual population prevalence for the PPV calculation to be right. So for example, if you do a case control study where you've selected a 2-to-1 ratio of cases to control, you have made the population prevalence, you fixed it by designing the study in that way. So you can't then go and calculate the PPV and the NPV in that study unless you do it using these things. So the PPV and the NPV are affected by your population prevalence. So if you use the study, if you want to estimate the population prevalence from the study that you've designed, then you can't use a case control study. You have to do a cohort study, and you have to make sure that it is a representative cohort study in order to get a correct estimate of this population prevalence. If, for example, however, with a case control study, you can actually estimate the accuracy. So true positive fraction, false positive fractions can be estimated in a case control study. So you could, for example, do a case control study, estimate your true positive fraction and your false positive fraction. Maybe you have some external estimate of the population prevalence, and then calculate your key. If your interest is in these PPVs and NPVs, then you can calculate it that way. But it's important to understand that population prevalence does affect these calculations. So it makes a real difference when you're thinking about what's the study. Then you have to pay attention to study design, much more attention to study design for these measures. The true positive fraction and the false positive fraction, you do need to pay attention to study design as well, but you have a bit more flexibility in the types of studies you can accurately estimate them in. Okay. So, oh, I see a question. Will using the population prevalence bias PPV, for example, if current measures underestimate the true prevalence? Yes. So I think that's the kind of the key thing about the PPV and the NPV. You actually have to have the right estimate of the population prevalence. So for example, if whatever measure you're using it does underestimate the population the true prevalence, then you are actually going to have a biased estimate of so if it's that's that I would say that's kind of the drawback of a PPV and an NPV. It's a more useful measure because it tells you in the context of clinical application. It's a very direct measure for a clinician trying to make a decision, but it is affected by having an accurate measure of your population prevalence, which is not a straightforward thing to do. Okay, so receiver operating characteristics curves or ROC curves. So we talked about a simple case of you have just test positive or test negative. Most of your biomarkers actually a lot of the time are continuous. And so how do we evaluate a continuous biomarker? A receiver operating characteristic curve is one of the ways we do it. So we're using Y is now a continuous biomarker. By convention, I'm just going to assume that higher values of Y are more indicative of a disease. If this is not actually true on the original scale, we can use a transformation to make sure that it's true. So for example, let's say you have a biomarker where low values are indicative of disease, you just take the negative of it. Or whatever transformation. You could do max minus the Y. As long as it's a one-to-one transformation, your ROC curve does not get affected by the calculation. It is invariant to these one-to-one transformations. Clinical decisions, however, are often made on the dichotomization of a diagnostic test. So it's sometimes you can actually use a continuous biomarker, and maybe that's actually something that, but eventually at some point you make a decision to intervene or not intervene, to do something or not to do something. So it's actually at some point it becomes a dichotomization. You might have like a risk prediction that you want on a more continuous level that allows you some granularity. So you're not being told use this threshold or that threshold, but at some point you're making a decision of what to do. That's how these definitions are now defined in. It's taking this continuous biomarker and making a decision. So now you're saying, like with a threshold C, if your continuous biomarker is it's greater than or equal to your threshold C, that's your test positive. And so what's the probability of having a biomarker at above or threshold C among those who have the disease? So we have equivalent basically definitions. When your biomarker is below the threshold, then that's a test negative. And so, and the false positive rate, similarly defined. Positive predictive value, same thing. What's the probability that among those who have biomarkers above the threshold, at or above the threshold, what's the probability of having disease? Negative predictive value, among patients with biomarkers below the threshold, what's the probability of being disease-free? So we have equivalent definitions that are based on dichotomizing your continuous biomarker. And your ROC curve is essentially the points of all possible dichotomizations that you can do. So all possible thresholds will then give you your ROC curve. And so literally what you're doing is you're coming up with, okay, in this data set, what are all possible, you think the definition is based on like negative infinity to infinity. Obviously, you don't need to evaluate such a wide range. You usually just go through the actual levels in the biomarker, look at all possible dichotomizations, and you come up with an example ROC curve like this. So this is an ROC curve for a couple of early detection of HCC biomarkers. Just a couple of things. So you can see here that they actually report marker AUC. AUC is the area under the curve. It is often used to summarize this entire curve. So let's say here they're comparing a couple biomarkers, DCP, AFP, AFPL3, and a way of summarizing what you're seeing in this entire ROC curve to compare these biomarkers is to then look at the area under the curve. And we'll get into why just now. But essentially that's one summary measure. A partial area under the curve might be actually more useful. And we'll get into that a little bit more. The other thing to point out here from this, this example is you can see here this like straight line. This is actually where a limits of detection issue. So AFPL3 is a biomarker that at this time of evaluation had a very high lower limit of detection. And so after a certain point, you basically, what you can't get is you could not get very high specificity because of that. And so that creates a bit of a problem. And so these are the kind of, when you see this, when you create these ROC curves and you see these like straight lines, that's actually limits of detection issues that you're dealing with there, okay? So why does AUC, what is AUC and why? So an ROC curve is telling you how good is your classifier. And if you have just a random classifier, so flipping a coin to decide whether a disease or no disease or cancer, no cancer, you would get an ROC curve that lies on the diagonal. So your ROC curve is your false positive rate against your true positive rate. And if it's the same as flipping a coin, it'll just go on the diagonal. If you have an absolute perfect classifier that is able to tell you everybody who has the disease or doesn't have the disease, you're going to lie over here. You're going to have a true positive rate of 100%, and you're going to have a false positive rate of 0, and hence a specificity of 100%. This does not exist, but it kind of gives you what the actual ROC curves that you get are somewhere in the middle. And the closer you are to this top corner, the better the biomarker is. The closer you are to this random classifier, the poorer the performance of the biomarker. So you can see why the area under the curve is a useful summary measure, because the AUC of a random classifier is 0.5. It's this unit square, and then it's half of it, so it's 0.5. The AUC of a perfect classifier, if it's essentially, it's going to go like this on the axis, is 1. So most AUCs are between 0.5 and 1, and the closer you are to 1, the better it is. So if we go back to our example, you can see here, AFP, this red line is a biomarker that had the highest AUC, 0.8, DCP with not as good with 0.72, AFPL3 essentially actually because it has this limit of detection issue in this version of the assay was 0.66. And then they also found that if they combine biomarkers, if they combined AFP and DCP, then they did a little bit better than AFP alone. So this is an example of like using ROC curves to evaluate these biomarkers. Just a little bit of technical details. Essentially, what we often do with estimating the ROC curve, oh, sorry, here's a question. Is there an AUC threshold for a good biomarker? I would say it depends on the context. Most of the time, when I'm evaluating a biomarker, I want to know what is the context in which I'm applying this biomarker. So for example, in this context of HCC early detection, the existing biomarker is AFP. So when I want to evaluate a new biomarker, in this case, DCP and AFPL3, I'm comparing it to AFP. So when there's an existing biomarker, we want to be able to beat the biomarker. So if we know what the AUC of this the existing biomarker is in the context, then we want the AUC threshold for when we're designing the study is this existing biomarker's performance. So AFP here in this context was a 0.8. So we had a very high bar to beat here, which they didn't. If you do not have, let's say this is an area where there's no existing biomarkers, and the current practice is is not based on anything, then you could actually even base it on AUC having a significantly better than 0.5. You could design your study so that am I doing better than a random classifier? So it truly just depends on the context. If there's nothing if there's nothing in the current practice, then we can compare it to a random classifier. But as soon as we actually have something, so you could actually have, let's say, clinical covariates that are used to make decisions. And so in that case, you want to then compare it to those, like, how do those clinical covariates perform? So it truly does go back to what I was saying, is understanding the current clinical practice. We then evaluate the biomarker in that context. These ROC curves are actually, we use empirical estimates. So that's actually, most of the time we use these non-parametric empirical estimates. So we're not making any kind of distributional assumptions. There are versions of the ROC curves that use, like, binomial assumptions and things like that. But most of the time we're actually just doing an empirical estimate of the ROC curve. So essentially these true positive fraction and the false positive fractions are proportions. We count the indicator this is an indicator variable that is 1 if the biomarker is above the threshold, 0 otherwise. So essentially what we do is we count how many times is the biomarker above the threshold in the patients who have the disease. So let's say ND are the number of patients in the cases, those with the disease. ND bar is the ones who are disease-free. So among those with the disease, we calculate how many, we do the proportion of how many have a biomarker positive. Among those without the disease, what's the proportion with a biomarker positive to get the false positive fraction. And then one minus that is our specificity. So just a little bit of a technical detail. It's a very simple calculation. So that's kind of key, that essentially true positive fractions and false positive fractions are proportions. Obviously, we don't do this manually. I do it in R. There's tons of different ways of doing it in software and things. Here's an example for those who are interested in how to do it with R. This is a data set that is from the Pepe's Books website. And so this is an example with pancreatic cancer. So you have, this is a diagnostic biomarker. So you have 90 cases with pancreatic cancer, 51 controls with pancreatitis. So they have like a condition, a pancreatic condition, but it's not cancer. You want to be able to differentiate those with cancer from those with pancreatitis. Here, I'm just literally just making this up. I actually don't know the context of the study, but for example, let's say you want to do a rule-in test. And we're going to control the false positive rate at 20%, so specificity at 80%. So not very high, because maybe patients with pancreatitis, they have some kind of a disease, and maybe the follow-up test is actually just an imaging study. So we'll allow more false positives. And then what we can do is we can estimate the ROC curve of two biomarkers, CA 19.9, CA 125, and at 80% specificity, 20% false positive rate, we get an estimate of the sensitivity, and we can compare them, and we can see that the CA 19.9 has a significantly higher sensitivity at the specificity that we're interested in. So I'm not going to go through all of this R code. It's more here for reference for people. You could also do a DeLong's test to compare the AUCs. And so here you see the AUC for CA 19.9 is 8.86, AUC for CA 125.71. And when you do a DeLong's test, your p-value is 0.006. And so pretty significant, but you could see that even just looking at these curves too. So just a little bit of R code for the for references here. Okay, so we're at the 1 hour mark. I'm going to pause here and let's take a 5-minute break so that people can maybe step away, take a break, ask some questions if they want. And we'll come back at 11:05 and start again. If anybody has any questions or things they want to discuss, feel free to unmute yourself and we can chat. Okay, so what is the statistician's role in the stage of biomarker development? I understand the statistical evaluation part, but for biomarker development part, it looks like it's very rigorous. It's for target population, target clinical practice. And we also need to understand the consequences of screening positive, like the ovarian example you provided. It seems the clinicians with high-level domain knowledges are more able to do so. Not sure how the statistician plays some role in it. I would say that this is that the statistician's role should be as early as possible and within a team. So for me, this is an area where being part of a team that actually understands every aspect of it is critical. So what do I mean by this? Let's say we're talking about an assay that is being developed in-house in a research lab. Your team needs to include the people who are developing that assay, who's studying the assay, who are running the assay. It needs to include the clinicians, like you say, who have the domain knowledge. How are we going to be using this assay? And it needs to include the statisticians so that you can understand fully. From our perspective, you need to understand this biomarker in order to study it and its clinical context and design these studies and analyze it. And I think from this, the lab and the clinical perspective, you need your statistician involved because you want to introduce as much rigor into your evaluation of your biomarker as early as possible in development. Because like I was saying about the ideas of randomization and blinding when you're running these assays, the more you can introduce these rigors, the more likely you're going to move a biomarker forward that actually has clinical potential, and the less likely that you're not going to be going down paths that actually don't turn out to be useful. So I would say that this is actually, I prefer, and I would say the most successful studies have been one where it's a team-based approach. Everybody who is a stakeholder in it is involved as early as possible, and that includes your statistician as early as possible. Sometimes, like, I'll be saying that, can we introduce more, like, rigor and things early? And sometimes it's not possible because it's like, maybe we're using a convenient sample to do our initial discovery, and that's all we have. And we need some level of evidence before we can propose to do it in the population that's closer to where we intend to use it. So, but understanding that and knowing that and having those conversations is just, I would say, critical to making sure that we have a biomarker that's moving forward and actually will progress in development to clinical use, hopefully. The statistician, when we talk about the rigor, like I haven't been involved in as much from the assay development point, but there's a lot of analyses that you do on the assay when you're doing the analytical development, for example. So that's also critical. I have done a little bit of that in some early detection biomarker studies. And so you do want to have as early as possible a team-based approach. And I would say, having a clear goal of where you want to end up for everyone is really critical, because if you understand where you want to use your biomarker, what's your intended clinical application, you can design your studies to get you to that point. If you don't have that clearly defined, it's very hard to design those studies and get on that trajectory. Okay, so how do we design these studies? So because when I was putting this is the first time I'm teaching this content. So I was either going to be on time or it was going to be way too much content. I think I'm heading a little bit too much content. But just to I'm not going to go through the Stata code for this. In the Pepe book, they actually provide Stata code that is simulation based. I think that, it's a fairly straightforward kind of program to use. If anybody has any questions about how to use it, just reach out to me. But you could also write your own simulation if you did that, if you prefer. They do have asymptotic calculations, but even they say don't use them. So the large sample theoretical results are not actually advisable. You want to just do a simulation study for these designs. So For example, here's an example when you have a dichotomous biomarker. When you have a dichotomous biomarker, what you need to be able to specify is your minimally acceptable true positive rate. So what is the lowest true positive rate that is considered acceptable? And your maximally acceptable false positive rate. What's the largest false positive rate you're willing to accept? And then this defines your region of your null hypothesis. And so then once you, the sample size calculation is based on rejecting the null hypothesis based on a confidence interval for your estimate. And so you then need to specify your anticipated true positive rate and false positive rate. You can then get a confidence interval. And if your confidence interval is within the, is outside of this null hypothesis region, then you can reject the null hypothesis. And so we specify for power, we specify type 1 error, and we can then get a sample size calculation based on that. So with a dichotomous biomarker, we're thinking of both the true positive rate and the false positive rate, setting our thresholds for that. When we have a continuous biomarker, you can design the study based on an AUC. I tend to not do that. And the reason is because I prefer evaluating the biomarker in the context I want to use it. So an AUC does tell you, okay, does the biomarker, like, am I going to get a biomarker that has higher accuracy than maybe the existing biomarker or something like that. But it's not necessarily a direct measure of its clinical utility. So I prefer actually designing the study to either do a rule-in test or rule-it-out test. So for example, when you're doing a rule-in test and we want to control our false positive rate, then the way I design these studies is I fix the false positive rate at its maximally acceptable value. And then I'm basing the study, the design on being able to detect a true positive rate that is clinically useful, that is significantly better than some minimal true positive rate. So for example, here we have a false positive rate that's maximal acceptable false positive rate of 20%. And let's say you have an existing biomarker that has a true positive rate at the false positive rate of 20% of 0.6. So then what we want to do is be able to beat that existing biomarker. And so that's how we would design the study. Can we do, for our rule-in test, can we get a higher true positive rate corresponding to 20% false positive rate than the existing biomarker? So I prefer designing studies like this. There are, you can see in this code, so you can design it based on AUCs, but I find AUCs are not as direct as how are you using an evaluation. Sometimes you don't have a choice. Maybe it's the context of you just need in the early phase studies, you might just be wanting to show AUC improvement. But once we get into validation studies, as we're moving up in levels of rigor, you want to then get to this either like a rule-in test or a rule-out test, for example. So a rule-out test would then fix the true positive rate at some minimally acceptable value and then estimate the false positive rate and make a decision of whether you've seen a significant improvement in the false positive rate. And again, you don't want to use the large sample theoretical results. There are simulation-based programs already available in Stata, or you can write your own if you prefer. So just a reference for others to use. This is a way the Pepe, Margaret Pepe's group has actually developed a program. You can then load it into Stata from the command line. And then here's an example of how to design a study where you're testing the AUC. Here's an example of a rule-in test and a rule-out test. So you can see here, this one actually ended up being, I have to run this code maybe. I can't remember. So NSIM tells you the number of simulations. So these are definitely simulation based. This might be just using the default number of simulations and that's why it's there. So anyway, like I said, we're not going to go into details now. If you want to use it and want to see how it goes, reach out and I can help you troubleshoot it as much as possible. Also, just kind of we've talked about the ROC curves for disease status is yes, no. That's often not the case. We've actually just defined it that way. There are other ROC curves. For example, you have time-dependent ROC curves where your disease status is has changed, and so you basically have time to event data, and so your disease status is defined. So you have another level of like the time points. So now you have D of T is 1. So if you have been, like, let's say if your failure time is by lowercase t. So let's say this little t is 1 year. Did the patient develop cancer within 1 year, or after 1 year of follow-up, are they cancer-free? So these are time-dependent ROC curves are defined in this context, and you can look up, I have provided a reference for how to estimate these when you have censored survival data and a diagnostic biomarker. There's a whole area of literature that would be a whole other set of training courses for that. So I'm just giving you a little bit of a heads up, but we're not going to any of these details. The other thing to kind of go into a little bit is that we talked about, like, you know, biomarkers, and we talked about a single biomarker. It's actually very unlikely, like, you know, so we often evaluate single biomarkers at a time, but if you want to actually have clinical utility, you're probably going to end up combining biomarkers, maybe even combining them with clinical information to actually improve the classification accuracy. Maybe you want to understand the association between your biomarker and your outcome adjusted for important clinical covariates. And so we use regression models for that purposes. If you have a binary outcome, then we're using logistic regression. If you have a time to event outcome, we're using Cox proportional hazards. I haven't really used them so much, but you have tree-based methods. So all kinds of like methods basically for regression with so combining things with the idea of making classifications. And so a lot of these kind of this is like, you know, existing methods, how you do this, model building, all of that kind of stuff still plays when you are doing biomarker evaluation. They're still all important, looking at model fit, model checking, things like that. The caveat I want to introduce here is that building a model and evaluating, let's say you come up with, you combine these biomarkers, you combine some clinical information, you come up with like, okay, this is going to be my score that I'm going to use to make a decision with. I come up with some kind of some kind of a score. If you build the model and evaluate it, let's say you come up, you look at the ROC curve of that resulting score, you are overestimating the performance of the classifier. Statisticians in the audience will know this, but it's key for everyone to understand this. There are techniques for internal validation. You can do cross-validation, you can do bootstrapping, things like that. But if you just do a straight regression model and then use the score and calculate the ROC curve, you have overestimated the performance of your classifier in that ROC curve. So it's a heads up that when you want to combine biomarkers with clinical information and things like that, then you're increasing, like, okay, then you need external validation and things like that to actually evaluate the performance of whatever score that you came up with. Some other kind of just things as a heads up, I wanted to make sure that everyone's paying attention to. Let's say if you're doing prediction, like you come up with, you want to be then look at the calibration of your score or whatever. So the difference between the observed rate and predicted rates. So let's say you have a biomarker and it's predicting something. You want to look at what's actually the calibration of that biomarker or that score. You want to look at improvements in prediction. Let's say there's an existing one, then there's tools for that. Let's have classification tables, standard net benefit, net classification index. I would say I put that with caution because there is some issues with the net reclassification index. And like I said, we want to assess goodness of fit. So all the things that you would need to pay attention to when you're doing model building still apply in the context when you're doing biomarker evaluation. And so those all need to be paid attention to. Okay, so let's get into a little bit of clinical trial design. So I'm using the JCO paper from Dan Sargent as my reference for this. And I think it's a very useful paper, so I would encourage everyone to read it. And so this also gets into the question from earlier, what's the difference between a prognostic and a predictive marker? So here's an example. So the clinical trial design for biomarker validation. So now we're getting into the context of you've been diagnosed with cancer, and now we want to design a clinical trial. So there's a therapeutic aspect now for these biomarkers. And so then it depends on what type of biomarker. So this is an example, two examples of prognostic biomarkers. Prognostic biomarkers informs you about the outcome irrespective of your systemic therapy. So for example, patients with a tumor expressing a specified biomarker have poorer survival compared to patients without the biomarker. So here's an example of when the marker is high, you have poorer, your median survival is lower, regardless of whether you received no treatment, treatment A or treatment B. And in this context, treatment A and treatment B don't make any difference. And here your hazard ratio actually for the biomarker is 0.5. So when your biomarker is low, you have longer median survival. And when your biomarker is high, you have poorer median survival. Here's an example with the same thing. This is a prognostic biomarker. Your hazard ratio actually still here is 0.5. You can see median survival is basically half whether you have treatment A or treatment B. But this biomarker doesn't tell you whether you should get treatment A or treatment B. All it tells you is that if you have this marker, if your marker is high, you have poor prognosis. You have lower median survival, whether you received no treatment, treatment A or treatment B. So it's not, a prognostic biomarker is not being used to guide your choice of therapy. It is being used to just describe what is the prognosis of the patient, irrespective of your therapy. And often we actually have biomarkers that have predictive aspects and prognostic, and then that gets a lot more complicated when we're evaluating them. A predictive biomarker predicts the differential efficacy or benefit of a particular therapy based on the biomarker status. So here's an example. When you have no treatment, median survival is 6 months. When you have received treatment A, whether your marker is high or low, median survival is 9 months. But when you receive treatment B, if you have marker status low, your median survival is 18 months. So you have a hazard ratio of 0.5 in treatment B when you have marker status low. So this is telling you that you can use this marker to decide whether you should be received treatment B or not. Here's another example where this is actually both a predictive and a prognostic biomarker. So when your marker is high, you can see across the board, they have lower median survival, but you also have a differential effect of a particular therapy. So your hazard ratio for comparing high versus low in the no treatment and the treatment is 0.5. Your median survival is basically half. But if you receive treatment B and you have biomarker low, you actually have a benefit with treatment B. So your median survival goes from 4.5 to 18. So this is telling you that not only is it prognostic, it's predictive. It's telling you differential efficacy of treatment B in patients who have low biomarkers. So it can be used to guide your choice of therapy. And let's say we want to be able to evaluate this in a retrospective analysis, then the key is when we have a predictive biomarker, it means we have a significant interaction between the biomarker and the treatment. Okay. So before designing the trial, again, we want to think about as we said, talked about earlier, no matter the context in which you're studying the biomarkers, there is like key things we need to understand the context in which we're evaluating biomarkers to design these studies correctly. Sorry, I'm just moving this thing so I can see if, okay. So the relationship between the outcome and the biomarkers need to be thoroughly explored in a retrospective analysis before you design a prospective trial. So like I said before with the early detection, same principles apply. You want to thoroughly understand your biomarker, the confounding factors, the assessment of, understand the confounding factors, etc., in order to design the correct study. So you don't want to just go straight to a prospective style. You want to, it's a prospective clinical trial. You want to do a thorough evaluation of your biomarker and retrospective analyses so you fully understand it before you plan your prospective clinical trial. But there is a caution that you need to be careful about what retrospective studies you're using to evaluate your biomarkers. And if it was, let's say, if you had like a clinical trial that you then had bank specimens and you go back and evaluate it, if your outcome of like recurrence was and progression was based on like, you know, standardized measures and regular follow-up, then you can do that. However, let's say you have an observational cohort, then you have to be careful because your assessment of recurrence and progression may be biased in that kind of a retrospective study. So when you're evaluating your biomarker in that study, you need to understand those biases that are present because that could affect the evaluation of your biomarker. So you do want to do these retrospective evaluations, but you just need to be very careful when you do them in this context, especially. You must be able to propose a specific testable hypothesis. So basically going back to what is the intended clinical use of your biomarker, and and then design your hypotheses to test that intended use. You want to understand your biomarker. Is it specific to the cancer of interest? Is it present in the other disease types? Again, target population keeps coming up every time throughout the literature in all of these contexts. We need to understand the population, and you actually, especially for these clinical trials, because you're going to see how large these sample sizes are to actually design these predictive trials, biomarker trials, you want to make sure it's going to have substantial clinical relevance once you start going down this path. So that is kind of key to keep in mind, that you want to make sure, like, what is the existing landscape of treatment maybe for this biomarker? You don't want to necessarily go into this where everybody does really well in this population. It's going to be very hard to make an impact then. So that's kind of a challenge, I would say. You need to have an estimated prevalence in the target population of the biomarker. So that's another area. Being able to understand, to design these trials, you need to know in your target population, what's the prevalence of biomarker positive versus negative, for example, because that is critical for the design phase. And it's not actually always a straightforward thing. It's not always easy to estimate, and it's not always known, but you do need it. Or if you make an assumption that's wrong, it can really affect your trial. The evaluation of biomarker, going back into it, method of measurement is reliable, and the specimen to measure the biomarker is available and accessible. The other thing I would come up to is, because this is now talking about prospective studies, it's also key to, lots of times when you do the retrospective studies, you want to make sure that the evaluation of the biomarker from these retrospective stored samples is the same as the evaluation of the biomarker when you have, let's say, a fresh sample. So that's also a key thing to think about, and going back to the same principles. Some of the primary endpoints you could come up with, let's say in the neoadjuvant setting, you could have response rates. So for example, pathologic complete response. In the adjuvant setting, overall and metastatic setting, overall survival is the gold standard, but it may not be feasible if it ends up being that the trial duration is so long that the relevance is compromised. Let's say you have to do like a 10-year study, and the field has moved on by the time you finish your study, then that might not be feasible. So then you have options like disease-free or recurrence-free survival, progression-free survival in the metastatic setting, or response rates. So sometimes what you might not be able to do is your key, like, your intended trial. What you maybe need to do is a series of trials to get you closer to your intended trial. So the one way we can design these studies is to do an indirect assessment. So here what we're doing is we register patients, we test the biomarker, but within each marker, let's say marker level negative and positive, you still just randomize patients to either treatment A or treatment B. And so the way we can then evaluate the study at the end of the design, either you can design the study to do separate tests within each marker group. So for superiority, So in those with marker level negative, is treatment A or treatment B? Is treatment B, let's say treatment B is your targeted therapy, and treatment A is standard of care. Is treatment B significantly better than treatment A? In marker level positive, same question. Alternatively, you can design the study to just do the interaction test. So you can design the study to say, is the treatment effect different in marker level for marker levels negative compared to positive? That does then, usually then, if you have that significant interaction, that tells you that there is a differential treatment effect. You would have to power the study if you then wanted to do the separate tests. We'll kind of get into that when you see the numbers, the example they gave. But you can go it either way. If, for example, you do separate tests, then you actually haven't powered to study the interaction. And so that could affect whether your ability to draw a conclusion. So it depends. Another alternative way of designing these studies is for direct. So you can see over here that the marker level is being measured prior to randomization, but it's not actually affecting any treatment decisions. It's basically, you're still randomizing to make a treatment decision. A direct assessment trial design is randomizing to either a marker-based strategy or a non-marker-based strategy. And then when you have a marker-based strategy, you're using the marker to make the treatment decision. So they recommend that the marker status be determined prior to randomization to ensure that you have availability of the biomarkers in all the randomized groups. I would say you'd have to have, you may want to make sure everything's blinded, for example, but you want, this is to ensure a completeness of your data at the end of your trial. But so the first option for their design is in the marker-based strategy, everybody who's negative receives standard of care. And in the positive biomarker, they'll receive, let's say, this targeted therapy. And then, but then in the non-marker-based strategy, everybody just receives standard of care, which is treatment A. So in this design option, what we then will do then at the end of the study is compare the outcomes in the marker-based strategy to a non-marker-based strategy. And so if using the biomarker to make a decision on treatment is useful, you'll have better outcomes in your marker-based strategy than giving everyone just the standard of care. However, this option A does not tell you about if you have a biomarker that is prognostic. This option A does not tell you if you have a treatment that is just superior regardless. Because you didn't treat anybody in the non-biomarker based strategy with treatment B, what you could end up is just say, let's say treatment B is just a better treatment overall. You could falsely determine that, falsely attribute that to marker level positivity, like being the marker-based strategy, when actually the treatment B is just better. And so that's the caveat for this design. So the alternative that they propose is to do a randomization in the marker-based strategy. And I think this is obviously a more logical approach, but it does increase the number of patients because now you have more arms and things in your trial. But you then either make a decision, you either do a marker-based strategy for your clinical design, or you do a non-marker-based strategy and you just randomize patients to decide whether you should give them treatment A or treatment B. And same thing at the end though, you're comparing a marker-based strategy to a non-marker-based strategy, and you compare the outcomes, and then that's how you evaluate your approach and your predictive biomarker. So the way to actually design the study and to figure out your sample size would be, I would say, to use a simulation study. I think you might be able to come up with some, I don't know whether there's any software that does an interaction, for example. How do you, I'm sure they have come up with it, but I find a simulation study to be the most useful. Some of the information that you need to collect, you need to, like I said, you need to know your marker prevalence. That's important to understand. You want to get a hypothesized benefit of treatment. So this is a table I think about, like when you fill it in, like these are the values. You want to know median survival in the marker negative, marker positive, unselected for treatment A and treatment B. So, and these are all related to each other and things like that. So you want to know how, hypothesize, how is this actually, this marker treatment interaction going to behave? And then we specify our power, type 1 error, and follow up. So the example they give is in colon cancer. And so you have a predictive biomarker, TS. Your treatment A is irinotecan plus oxaliplatin. Your treatment B is irinotecan plus fluorouracil. And your hypothesis is that treatment B is most effective in patients with low TS levels. So to design, it's also, though, the complication is that TS is a prognostic biomarker as well. Patients with high TS levels have poorer outcomes even in the absence of treatment. In the simulation study, we assume survival follows an exponential distribution. And here they're assuming a very high prevalence where 50% of the patients have high TS levels. And so here's the hypothesized treatment benefit. In an unselected population, median survival is 15 months, regardless. So irrespective of your TS status. If you have TS high, let's say when you think about treatment A, TS high has smaller median survival, lower median survival than TS low, and in treatment B, TS high has lower median survival than TS low. But you can see the differential effect that when you have TS low, you have, and you're given treatment B versus treatment A, you have an improvement in your median survival. When you have TS low and you're given treatment A instead of treatment, and treatment B has a worse median survival. So here actually what you call it, the treatment A is preferred in TS high and treatment B is preferenced in TS low. That's not always the case. Sometimes you just know that in certain groups, you wouldn't prefer one over the other, but you wouldn't necessarily have a preference in the other groups. So in the TS high patients, the hazard ratio for A versus B is 0.6. In the TS low group, the hazard ratio for treatment A versus B is 1.25. And so the hazard ratio for the treatment by biomarker interaction is 0.69. So if you do the direct assessment method and you do a marker-based strategy, to get that estimate of the median survival for a marker-based strategy, you actually have to use this conditional law of total probability. Anyway, you have to calculate what that it would be, taking into account what's the prevalence of the biomarker, but you end up with the marker-based strategy will have a median survival of 16.5, and a non-marker-based strategy arm, so this unselected, will have a median survival of 15 months. So if you do this direct assessment method, the hazard ratio that you're powering your study for is 0.91. So that's very close to 1. So that's going to give you a hint of how large the sample size is going to be. So if you're powering the study for this treatment biomarker interaction, you're looking for a hazard ratio of 0.6, interaction of 0.69. And so that gives you a smaller sample size. Most likely, you're going to expect a smaller sample size. And part of the reason why this hazard ratio is so high is that in the non-marker based arm, 50% of the patients receive an optimal treatment by chance alone. Because the marker prevalence is so high, you're just going to optimally give patients give the patients the optimal treatment by chance. So here's their sample sizes. And so let's just look at the number of events required. And you can see these are all very large. And so when you have like these kind of studies, you're actually, what you are doing is a prospective subset analysis. And so you're going to have very large sample sizes. And so let's say you wanted to do the separate tests. So compare the treatments within each arm. Then you're looking at the number of events required is 2,500. If you do the test of if you just power it for the test of interaction, you're looking at 1,200 patients. And then, like I said, with the modified marker-based strategy, that's the one where you're looking for a hazard ratio of 0.91. Oops. And so that's where you get into the 4,600 patients, 4,600 events, which is like 8,000 patients because this is per arm. So very large studies in this example, very large studies in this example. They do give an example of a case where you can get a smaller sample size by actually having the biomarker tell you not just who you should treat, but not who you shouldn't treat as well. And so then you can reduce the sample size. So it just depends on the assumptions, but this is kind of a caveat of like, these all seem pretty reasonable. And, you know, when you look at these are things that seem like something that, you know, a hazard ratio of 0.86 and 1.25, those actually seem like expected hazard ratios you would see in our studies. And you end up with very large sample sizes. So this is also going back to my principle of once you're actually designing these trials where you're doing an intervention, you're doing a prospective study and you're doing an intervention, you're talking about very large studies. And so you want to be sure that your pipeline of taking your biomarker from discovery to these prospective trials is a solid pipeline that includes thorough evaluation and rigorous evaluation, because you do not want to run a trial with 2,000 patients. That's the smallest one, about 2,000 patients. And it turns out you have problems with your biomarker. So just a summary of the clinical trial design part. Investigation of the predictive effects is a prospective subset analysis. And so a large sample size is often not avoidable unless you have like a really great biomarker that's going to really revolutionize the field and really split your patients into like, you know, the treatment groups. But most of the time we're making incremental changes. So you don't often have that. The marker by treatment interaction design uses all the randomly assigned patients to maximize the efficiency of the design, but it's not powered to test the treatment effect separately in the two subgroups. So you can say that I have a significant difference in the treatment effect in each in the biomarker status, but you are not powered to test that treatment effect separately. The marker by treatment design that uses separate tests may not provide convincing evidence of utility. So for example, let's say you design it for separate tests and maybe you have, you don't get statistical significance. Then maybe you might have an estimate that provides a hint of the direction, but you don't have statistical significance. So now you don't know where to go with this. You're stuck with that. So that's the caveat. The direct assessment design is more feasible potentially so even though, so you can see over here, the sample size for the direct assessment, this modified marker design is getting very large. But sometimes you actually might need to then do that, especially when the treatment decisions and the number of markers become more complex. So let's say you actually have like a panel of biomarkers, a panel of treatments, you're doing these like complex decisions. Treatment marker by treatment designs are not going to be feasible in that context. So then it potentially becomes much more feasible to do compare a marker-based strategy for designing treatment effect to a non-marker-based strategy. That's just the caveat of that. And then another question you can do is, to think about is, could you design your phase 3 trial to then do a retrospective evaluation of your biomarker as kind of your study? So if you design your phase 3 trial, keeping in mind and actually specifying that you're going to do this retrospective evaluation of your biomarker, you can get a very high level of evidence for the performance of the biomarker, but you just have to do that a priori. And so something to keep in mind with is to think about, like, can you combine it? And that might be a way of conserving effort and making it more feasible. What I haven't gone into, we mostly talked about single time point assessment of biomarkers. A little bit of a plug for my own research is when biomarkers are assessed multiple times, you could be doing this in risk prediction, early detection, monitoring for recurrence. And so then we start getting into a whole new field of the longitudinal trajectory of biomarkers and how you evaluate it, beyond the scope of today's training seminar, but I like to put a plug in for my research. So if anybody's interested in these areas, feel free to reach out to me, and I'd love to discuss more. But a lot of the principles we're talking about all for this whole process, when we're talking about single time point assessment of biomarkers, there are a lot of principles that apply. It's just that once we have longitudinal assessment of biomarkers, we have more information, and so to exploit potentially. So key takeaways from today's training. I keep saying this, but I think it's just critical. Context for the usage of the biomarker. You need to fully understand, and even as a statistician, you need to understand thoroughly how are you intending to use the biomarker? What's your end goal for how you're going to use this biomarker? The biomarkers that are most likely to succeed are ones where you have a very clearly defined end goal and you design your studies to get to that end goal. What stage of development are you at already? Understanding what data do I have? Where am I now is also really critical to then planning how am I going to get to my end goal, and how am I going to design my study to answer the primary question of interest. It's critical to thoroughly evaluate a biomarker before it gets incorporated into clinical practice. There are tons of cautionary tales of when biomarkers were not thoroughly evaluated and incorporated into clinical practice, and what can go wrong in that. And sometimes it's like, so here's an example, to a certain extent in my field, AFP is an early detection biomarker for HCC, and it is an early detection biomarker, but there's still actually, because there hasn't been a randomized clinical trial showing benefit, it's still a very controversial biomarker. And whenever there's guidelines, there's a question of, like, should we be doing it or shouldn't we be doing it? But it's actually very widely done. So when you do a study and you plan a prospective study, you have to use it because it is recommended in the guidelines. It's not required, but it's recommended. So it actually makes it, when a biomarker is in this wishy-washy phase, it actually makes it a little bit difficult in that context. So it's got utility, but it hasn't been validated in a randomized trial. What do you do with it? And so ideally, thorough evaluation of a biomarker before incorporating into clinical practice provides a much cleaner assessment. On the other hand, though, medicine is rarely clean, so we do the best we can with what we have. So thank you very much for all your time. Here's some references for what I use to put this presentation together, and I'm happy to take any questions. I hope this was useful, and gives everyone a good framework for doing a biomarker study and collaborating.
Well, it looks like everybody liked your talk, Nabihah. Oh, we do have one question in the chat right now from...
Are you using data for these simulations, or do you use R as well? I actually use R. I literally only use data to do when I want to use Margaret Pepe's program. So I would not be able to help you do a data simulation, but I do R simulations. That's my preference. But the data program is actually very easy to use. So sometimes I don't. If it's a very straightforward design, I actually just use the Margaret Pepe's software and just do it that. Do you have other ways to assess if a biomarker is predictive apart from an interaction term of a biomarker by treatment? You know, there are other ways that they get assessed, but I would say that they're usually just, they're not as convincing evidence. So for example, sometimes you do like a single arm study where you, with everybody with the biomarker gets treated with a particular treatment, and then you compare it to, like, you know, historical controls. So you're not doing an interaction design, it's like a single arm study. I would say that gives you some evidence, but it's not convincing evidence, I would say. It's not the level of convincing that will probably, like, you know, change practice, although sometimes just change practice anyway. But I would say it was not the level of evidence that a statistician would be considered to change clinical practice. And you could, for example, in the paper they had was treatment B works better in everyone. So if you had done the single arm design where everybody who has biomarker positive got treatment B, you compared it to the historical median survival, you'd say, oh, everyone with this biomarker should get treatment B. But actually, maybe everybody should get treatment B, not just those with the biomarker positive. And so you wouldn't have that. You wouldn't know that. So the highest level of evidence is the interaction term, but sometimes that might not be feasible. So if you have a single arm design to get you some like preliminary evidence that then gets you gets you closer to your confirmatory validation study, then you might end up doing that. But the interaction, I would say, is the highest level of evidence. Okay, I'm not sure if you have any experience on this. Sometimes for long-term endpoint, localized cancer, we want to find a surrogate endpoint. Is the surrogate endpoint somewhat like a biomarker? You know, I actually don't have experience with this. So, but it was something I was thinking about a little. So I noticed in the in the FDA guidance for biomarkers, they do talk about a surrogate endpoint as a biomarker. So I did not go into that because I think that's a whole nother level of complexity, because I think surrogate endpoints are important. And so you actually want to, I think that's a whole, they have, like, you know, complexities and subtleties that you need to pay attention to there. So I would say I don't have experience, but I do think that there is some kind of that the concept does exist in the field. I just don't have the expertise to answer that right now, but it is something that I saw as I was doing some literature reviews. So it's something that I'll pay attention to, and maybe we can chat about offline another time once I've looked into it a bit more. Okay, I think that's it for the questions. Last call for questions for anybody who's left. Okay, well, thank you everyone for attending.
Thank you, Nabihah.